All right, some more classic histology, classic anatomy, but at the cellular level. Last week we looked at the pancreas, we looked at the islets of Langerhans and all those exocrine cells and endocrine cells. We're still working our way through the GI tract, so another major organ associated with the gastrointestinal tract is, of course, the liver. It does so much work, the liver, that I'm not even going to try to explain all the things that it does. But let's have a look at the cellular arrangement of the liver, because it's a common thing in most anatomy syllabuses, and it seems complicated when you first look at it, but it's not. It's quite logical. I've got a bunch of liver slides actually, but I think we're just going to look at two of them. One, do most of our work with one of them, but we have to consider not so much what the liver does, but how it does it, or maybe a bit of both. Okay, liver. Okay, so imagine you're designing a factory and you've got two inputs. So the liver has got a hepatic artery proper, that's bringing arterial blood into the liver. So normal arterial blood, rich in oxygen and the usual bits and bobs that you find in arterial blood. That's the first input. The second input is the hepatic portal vein. Now the hepatic portal vein, it's a portal vein because it's carrying blood from one organ to another organ. It's carrying the blood from most of the gastrointestinal tract to the liver. You eat a meal, you ingest it, you digest it, you absorb those nutrients go into the blood of the gastrointestinal tract. Those veins drain to the portal vein and then go into the liver, because the cells of the liver are going to look at all that blood and do their 500 different jobs on some of that blood. So you've got two inputs. You've got your normal arterial blood, hepatic artery, and then you've got your blood from the gastrointestinal tract, hepatic portal vein. So you've got two inputs. All right. So imagine you've got a conveyor belt running through your factory. The cells of the liver are the hepatocytes. Makes sense. Hepatic refers to the liver, cyte is a cell. So almost the entire liver is just hepatocytes. So the hepatocytes are your workers, and they're going to work on that blood that's coming in. So you've got your two inputs. You're going to arrange your conveyor belts such that all of your workers, all of your hepatocytes can touch the conveyor belt. They can't touch the conveyor belt, they can't do any work. So you've got to arrange all your hepatocytes up against those inputs somehow. Now, that blood, those inputs are going to flow across the liver, so through your factory and out the other side. But you've got two outputs. One output is fairly straightforward. The blood that goes in from those two inputs is going to flow out through hepatic veins, and the liver is wrapped around the inferior vena cava. So that blood is going to flow out through the hepatic veins and into the inferior vena cava. So that's one of your outputs. But your workers, your hepatocytes, are also going to make bile. That's another output. But bile is going to be used in digestion. It's going to help digest fat. It's going to help with absorbing certain vitamins. So the second output is bile. So your workers, your hepatocytes, need to be able to put that onto another conveyor belt. You can't mix the bile with the blood, and that bile has to come out of the same part of the liver that your two inputs went into, because your hepatic ducts and then your common bile duct are going to go to the duodenum, so they're going to go back to the small intestine. So how would you arrange that? Your two inputs, your one output in there, and then your other output back to the small intestine, but you've got to keep everything separate. And then you've got as many cells as you can fit in here, so as many conveyor belts as you can fit in here. How would you arrange that? How would the liver arrange that? Let's have a look. The cells of the liver, so yeah, the cells of the liver, the hepatocytes, they have like 500 functions. But let's start recording. The cells of the liver, so if we think about what's coming in through the gastrointestinal tract, what you've absorbed, they're going to take those nutrients, they're going to store a lot of the nutrients, the carbohydrates and the proteins, the sugars and what have you. If you ingest alcohol and toxins, they're going to break those down. They're going to work with various vitamins. The cells of the liver are involved in recycling red blood cells, so they're getting rid of bilirubin and they're putting that into the bile. Of course, they're making bile, so they're making the bile salts and various other bits to help with fat digestion. And where's my tissue? There it is. Yeah, they're very, very busy cells. They're involved in all sorts. Okay, so let's have a look. Is that in focus? I only struggle to focus for the camera, my low power lens. That looks like it. This is my 4 times objective, and we've just got a, you can see the edges, look, it's just a square of liver from some random spot inside the liver. So pretty much everything we're looking at here, these are all hepatocytes. So if I zoom in a bit, it's very palely stained, this tissue, but oh look. So right, what we're looking at here, well, we're looking at mostly hepatocytes. These are almost entirely hepatocytes, but you can see the hints of channels. So you can see those conveyor belts running alongside those hepatocytes, and we can see some bigger circles. So we're starting to see some of those inputs and outputs. And we talk about central veins, and we talk about portal triads. So there's going to be 3 things at a portal triad, and that's quite good. You can kind of imagine or see there kind of a star shape of how all those channels you can see are leading towards that central tube, that central vein, and they're running from these other tubes, these circles, further out there. Right, let's actually have a look at this other section, which has got a bit more stain on it. I think might be a bit better for us. See what we've got here. Okay, let me walk you through the parts, the things that we can see, and then I'll join it all together to talk about lobules and what have you. But there's the wider view of this tissue. So we've got a sea of hepatocytes with lots of circles. So those are tubes, input and output tubes. If you look at that, can you imagine any shape or structure in there? Everything is arranged. Right, okay, so that's quite nice there. This will be our portal triad. This is where we have 2 inputs and 1 output. So if I zoom in again, okay, so we're looking at circles. These are tubes that have been cut in transverse section. That's why they look like circles. How do we figure out which is which? Well, that little circle top left, the cells around the lumen are, well, they're epithelial cells. They are as tall as they are wide. So they are cuboidal cells. I can go in even tighter, can't I? So on the left there, that's a very, very small tube, and there are cuboidal cells, and we can see to the top and the right there's another small tube, very, very small tube, with a simple cuboidal epithelium forming the epithelium that's lining that tube. Those are the bile ducts. Remember, we looked at the gastrointestinal tract and we saw that gastrointestinal tract, that tube that runs all the way through us, inside us, is lined by epithelium because it's an external surface inside us. So there's a barrier protecting the proper inside us from stuff passing through the gastrointestinal tract. So then the glands and structures that have grown from that gastrointestinal tract and that duct into that gastrointestinal tract are also lined by an epithelium. So if we're seeing a tube here that's lined by an epithelium, this is a bile duct which is going to pass bile into the small intestine. So these will drain to the hepatic ducts, then the common hepatic duct, and then the common bile duct, and then to the duodenum. And then in the centre there, we have a large tube with very flat cells, so that's an endothelium. And it hasn't got much of a wall, it's got quite a thin wall. So one of these is the portal vein, and one of them is the hepatic artery proper, as it were. So that's our portal triad, and of course we call it a portal triad because we expect to see a bile duct, a portal vein, and a hepatic artery. But because of the various branching and what have you, we're seeing two bile ducts and what have you there. So there's the portal triad, and then can you see, maybe you can see, don't forget, so that space around it is probably artifactual from fixing, probably from fixing and dehydrating the tissue. But if we slide over, what we're seeing there is we're seeing lots and lots of hepatocytes, and we can see channels between the hepatocytes. Can you see that the hepatocytes are pretty much no more than one row of hepatocytes thick? And this is what I was referring to when I said if your hepatocytes are your workers, they all need to be able to reach the conveyor belt. So the gaps between them will be the sinusoids, the channels that the blood is travelling through from the portal vein and the hepatic artery, and they'll be running across here. See if we can follow the flow. They'll be running to the central vein. There's a triad. There's a triad. There's a triad. There's a triad. And then, so, I don't know if I can just about get them in, but down here, this is the central vein. And all those sinusoids are leading towards that central vein. I don't know if you can get a sense of how that looks. But those sinusoids are all leading towards that central vein. So this is your hepatic lobule, that's the middle of it, and these portal triads are the inputs, and they're sending blood then through those sinusoids down to the central vein. And like I say, all those hepatocytes were arranged in kind of plates, a single layer of hepatocytes wide, so they can all reach into the conveyor belt, they can all reach into the blood and work with the blood. Why don't you have two layers of hepatocytes and a sinusoid on either side? Because then they could both reach into the conveyor belt. Well, that's because they're producing bile. So of your row of hepatocytes, on one side they're working with the blood, and on the other side they're putting out bile. So the bile canaliculi, the little canals carrying bile, are on the other side to the sinusoid. All right, if I zoom out, let's try and see these hexagons. So the hepatic lobule is your hexagon shape that is always drawn so nicely in textbooks, but you have to use your imagination a little bit to see them when you're actually looking at liver histology. Can you see there's a central vein in the middle? You can kind of see the sinusoids leading into it. You can kind of see where they're coming from. You can maybe imagine an edge. But let's zoom in. You got it? You got the hexagon? there's your central vein there. Hey, look, it looks like a double vein. Well, you know, so that's biology for you. And if I scroll up a little bit, you can see those canaliculi are flowing from, there's a portal triad on the left, and there's a portal triad on the right. The one on the right's not very clear because of the plane of section. You can just make out there are various tubes in there. If I go down to that central vein and then slide out that away, we can see, oh, can you see? So there's one central vein. There's the next central vein. Can you see how the pattern of sinusoids changes there? It links and overlaps, and there's kind of a line, there's kind of a wall of the hexagon there between that portal triad and the portal triad that there's a bit of there, and there's another central vein down there, and there's another portal triad over there, and there's another, yeah. Anyway, biology. Sorry, this is real life. But you can see how tricky it was to figure all this stuff out, right? And it's described very nicely. Sometimes it does look cleaner than this. So that's your hepatic lobule. Now your portal lobule is another triangle where you have three. Well, that's well, where you've got three central veins. You can make all sorts of shapes with your human brain, can't you? Can you see there, though? If I can focus this. You've got three central veins, and they're making a triangle. And there should be a portal triad in the middle. That's your portal lobule. Let's see if I can find a better one. Yeah, that's actually a reasonably good hepatic lobule there. The reason this is so complicated is because you're not just planning your factory in 2 dimensions on a flat factory floor. You're planning a 3-dimensional factory, and you're fitting this into a 3-dimensional space in an optimal manner. So it's even more complicated. But can you see right in the middle there, we've got the central vein, and hopefully you can get the sense that those canaliculi carrying blood are leading towards the central vein, and around the outside, so further away, we can see more circles that have been cut through. Those are the portal triads. And we can kind of make out 1, 2, 3, 4, 5, 6 corners maybe, and get a shape of that hexagon. That is your hepatic lobule. There is another way of thinking about this, which is the hepatic acinus, which is a diamond shape with 2 central veins at either end of the diamond and 2 portal triads at either side of the diamond. Do you see how there we've got, not in perfect condition, but we've got 2 portal triads at the top there and a central vein at the bottom, so they're making a triangle, or the bottom half of the diamond. And if we scroll up, there's another central vein up there. Just about get it all in. So this hepatic acinus, what this is considering is that there's the input. So you've got the hepatic portal vein bringing blood from the gastrointestinal tract in here, and you've got the hepatic artery bringing arterial blood in here, so oxygen and nutrients, things that normal cells normally need, and then that blood, of course, is flowing across to the central vein there. So the hepatic acinus is not just that side, it's also that side going up there, but it's considering how, as these hepatocytes, as they get further away from the arterial input, they're getting less oxygen. So this gets talked about as zone 1, 2 and 3. So zone 1 are these hepatocytes closest to the arterial input, and then zone 2 is in the middle, and then zone 3 are the hepatocytes furthest from the arterial input. So these are the hepatocytes that get the least oxygen. So that's what, if you come across hepatic acinus, that's what that's referring to. It's kind of confusing, but it works. So yeah, like I say, there's a triangle between those two portal triads and that central vein. And if you take that triangle and then add it onto that triangle, you've got a diamond, and that's your hepatic acinus. Those, I think, you know, those layouts can be confusing because they can be difficult to see. But what you should be able to do is let's go find a really good portal triad. Really, when you're looking at this, if you can identify a portal triad, if you can find a central vein, you'll see all those sinusoids with the blood in it running towards them, and then it, you know, then you can see what's going on in the liver. There's a nice one. Then you understand microscopic liver anatomy. Oh yeah, that is a nice one. Look, okay. So there's our portal triad. We've got 3 tubes. Oh, right, yeah, okay, we've got more than 3 tubes. Yeah, tubes branch. But you've got 3 different tubes in there. We can see the bile duct because that's got those epithelial cells arranged so beautifully around its lumen, around its inside. So you've got the bile duct there, you've got the simple squamous, you've got the simple cuboidal epithelium arranged around there, then a little bit of connective tissue holding it all together. And then we've got 2 small tubes lined with endothelial cells, one with a thin wall and one with a slightly thicker wall. So the slightly thicker wall is your muscular artery, arteriole really at this scale, and the very thin walled one is the venule. So this is where we've got our 2 inputs, our hepatic portal vein input, blood from the gastrointestinal tract, and we've got our arterial blood input, and then we've got our bile duct output. That's your portal triad. And then if you compare that to a central vein, so here look, we've got lots of circles, but they don't look like that portal triad. We haven't got a tube there lined by a simple epithelium. It just looks like a very thin-walled tube, a very thin-walled vessel lined with very flat cells, and those are the endothelial cells. So that's a vein, that's a low pressure vein, right? That's a hepatic venule that's going to drain to the hepatic veins. And the other thing we can see is we can see the sinusoids leading towards it. If you're starting to get the shape of the flow, brilliant. So the hepatocytes themselves then, what we're looking at here, these plates of hepatocytes, these rows of hepatocytes, on one side there we've got the sinusoids with the blood in, and on the other side we've got the bile ducts. So blood is flowing in one direction towards the central vein, bile is flowing in the opposite direction on the other side of the hepatocytes out to the portal triad. There is one other thing to talk about in terms of the blood and the sinusoids. We'll have a look at the canaliculi on another slide in a moment where they'll jump out at you and they'll be obvious. But what we've actually got in here is you've got the blood running through the sinusoids, and like the other blood vessels of the body, those sinusoids are actually lined by very, very thin endothelial cells. So you've got your hepatocytes and you've got your endothelial cells, and there is a space in between, and that's called the space of Dis. And I think we can just about make that out in here, right? Don't know how much imagining you have to do. But we can see, right? Can you see the canaliculi? And you can see some very flat cells there, endothelial cells. So the space of Dis is the space between the endothelial cells and the hepatocytes. And the hepatocytes have got loads of microvilli on them because that's really useful when you're well, exchanging things between the blood and the cell, because you need as much surface area as possible. The reason the space of Dis is interesting is because in here we find the stellate cells, or the Ito cells. The reason those are interesting is not because they store vitamin A, but because when the liver is injured and the liver heals itself, it's these stellate cells that can produce collagen. They can produce fibers to help with repair. But the problem is, with repeated repair of the liver, these stellate cells make too much collagen. They make too much fiber, and the liver becomes fibrous. Now, if you consider that a huge amount of blood passes through the liver, and all that blood is passing through these microscopic channels between these rows of hepatocytes that are 1 cell thick, if that collagen starts to form in the space of this, it's going to start compressing the sinusoids, right? You're going to lose this layout. You're going to lose all these channels, and it's going to become so much harder for blood to pass through the liver from the gut to the inferior vena cava. And in gross anatomy, we talk a lot about the portosystemic anastomoses. There are links between the blood vessels taking blood to the liver and the blood vessels taking blood back to the heart, to the inferior vena cava and the superior vena cava. And it's this microscopic anatomy, when this breaks down, when this becomes fibrous, the resistance to flow of blood through the liver becomes so great that the blood then flows in the opposite direction through those portosystemic anastomoses, and we get varices, we get distended veins in the lower esophagus, you've got portosystemic anastomosis in the anal canal, in the abdominal wall, in the posterior abdominal wall, and all over the place. But it's those stellate cells, those Ito cells, in the space of Dis that are responsible for that. Bile canaliculi, you say. Not easy to say. Right, okay, so that's our normal hematoxylin and eosin stained slide. This slide is labelled as liver alkaline phosphatase. Alkaline phosphatase does lots of things around the body. In the liver, alkaline phosphatase is most associated with making bile. So it's associated with the bile ducts. This is a black stain. So what we're seeing stained here are the bile ducts. So now you can see that central circle is not a bile duct, but you can see those other smaller circles. Those must be bile ducts because there's quite a lot of alkaline phosphatase labelling there. Let's go up again. What's this? 20 times objective. See what I mean? That's a blood vessel in the middle. But these little tubes down here, you can just about make out the simple cuboidal epithelium, and there's a black stain on the inside, so they are carrying bile away from the liver. And look in here. So whereas before we were seeing, most noticing, I think, the larger spaces, those were the sinusoids carrying the blood. You can see here, now stained in black, that the bile canaliculi are much smaller. Little tiny channels running from the other side of the hepatocytes. Yeah, look, that's quite good, isn't it? So you can see that the blood sinusoids have not stained, but those little thin black stains, those are the tiny, tiny, tiny canals carrying bile away from the hepatocytes and towards the bile ducts. And can you see now how the hepatocytes are in rows 1 cell thick? But those bigger spaces on either side, those are the blood sinusoids, and the bile is in tiny little channels. Oh dear. Anyway, the concept is probably as important as anything else. The liver has 2 inputs, an arterial input with a hepatic artery proper, and then blood from the gastrointestinal tract filled with digested and absorbed nutrients comes in through the hepatic portal vein. Those two inputs then mix, flow past the hepatocytes arranged in single rows, and the blood is in the sinusoids between those hepatocytes. They can do their work on the blood, and the blood flows through and into the central vein and out and back into the inferior vena cava. The hepatocytes then are also producing bile. They pass that out into the bile canaliculi, those tiny little canals which run in the opposite direction to the blood flow, back out to the portal triad and into the bile ducts. And in the portal triad then we'll find those two inputs, branches of the hepatic artery, branches of the hepatic portal vein, and we'll find branches of the bile duct going out. That's it for the liver. In terms of the hepatic lobule, that's the hexagon, that's the arrangement of those inputs to the central vein output and flowing into the centre there. Whereas the other descriptions are just other descriptions of the same shapes. So the portal lobule and that sort of thing. But I think if you get the main features and you get the concept, you're 95% of the way there. Okay, I thought that was going to be more complicated than it was going to be, and then it turned out to be quite complicated anyway. Liver, done. See you next week.